Okay, we're going to get going, and this is the course on redox biology, and we've done this course, we do it every other year, so I think this is the 5th time we've done it, and it's a very unique course because redox biology, in the whole of US, there's only one university that has a redox biology course, and that's the University of Iowa. So this is very unique. And Dave Wink and myself, we work at NCI, but redox biology, it's important in many fields, such as we'll see the Heart, Lung, and Blood Institute is very interested in this, NIAID, NINDS. So redox biology, it covers many fields, and then my boss is Jonathan Wiest. He's in the Office of Training and Education at NCI. And he's very important because he helps fund this course. And so this course, we not only have the lectures, but we have the video cast. We video cast live to NCI Frederick, where about 1/4 of the NCI fellows are. And then 2 days after the lectures are all archived. You can watch it live on your computer on Mondays, 4 to 6, but then 2 days later it gets archived and put on YouTube, and you can call it up on your computer anytime. So we do several courses. One course that we're doing now is translational research in clinical oncology. That's Monday afternoons, 4 to 6. And the redox biology, as you know, is Tuesday afternoons, 4 to 6 in the fall. The redox biology class, it goes for 6 weeks. So we have 12 total lectures. The Traco course is much bigger. It goes for 13 weeks. And these are our main courses in the fall, and then the rest of the year we have other courses, such as a statistics class, we have classes in grant writing, classes in teaching. I'm also a professor of biochemistry at George Washington University. So the first lecture, I'm going to introduce the topics, and then we have Dave Wink, our resident expert in nitric oxide. He's going to be talking about chemistry today, and then biology next week. And we'll have Dave Roberts talking about angiogenesis. I come back and talk about signal transduction, and we talk about redox physiology. We talk about cancer epidemiology and inflammation in cancer. We talk about cancer therapy. Lisa Ridnow talks about redox enzymes. I'll be talking about some of those today. And we have Tom Lato from NIAID talking about NADPH oxidases, and Murali Krishna talking about cancer imaging. So the course is free, the price is right for all of you, and we mentioned that we video conference live to Frederick, but then it's video cast as well, so you can call it up at the NIH videocast website, which is shown in green. And then we also have a web page. And what we do is we post the PowerPoints on the web page once they're 508 compliant. And then this course is too big to take attendance. So what we do is we have a final examination. If you want to obtain a certificate, it's a multiple guess exam, 12 questions, you need to get 70% right to pass. So that's posted on our webpage at the end of class. And one interesting thing is right in the middle of our registration, they changed the webpage. So it used to be a different link, but now this is the correct link. Any questions on the organization? Okay, we're going to get going then, and we'll be introducing topics, reactive oxygen species, reactive nitrogen species. We'll be talking about hydrogen sulfide, carbon monoxide, and hydrogen gas. So these are all of our redox species. And the first one we go into is oxygen. We live in an oxidative environment, and that can create oxidative stress. We breathe in oxygen into our lungs from the air. We exhale carbon dioxide, CO2. And then the oxygen, if you add an electron to it, you can then generate the superoxide. So this is what Dave calls an instigator, as well as hydrogen peroxide. You add another electron, you add 2 electrons to oxygen, you can get hydrogen peroxide. This is also an instigator. And then it could be metabolized into the hydroxyl radical, which is sort of a deadly species. And I'm very familiar with ozone. I did my graduate training at Caltech in Pasadena, California. And Los Angeles has a tremendous amount of air pollution. And then around 2 o'clock every day, the toxic air would move from the coast inland. It would get to Pasadena around 1 to 2, and you'd breathe in and your lungs would start hurting because all the nerves were getting oxidized by the ozone. And ozone in the lower atmosphere is bad. It's about 0.6 per million in ozone. That's when you get bad pollution. But then the pollution would keep moving east, and it would go to Riverside about 5 p.m. So about 4 o'clock, you started feeling better in Pasadena. And the saying was, for every 2 years you live there, you lose 1 year of your life because of the severe oxidative stress on your lungs. But ozone in the upper atmosphere is good, and it protects us from UV light. And a problem is that the ozone in the upper atmosphere is decreasing. Hence, in some states like Arizona, Texas, Florida, they're getting excessive ultraviolet radiation, and the melanoma rates are increasing in these states dramatically. So whenever I go out in the sun, I wear sunscreen. And then additional oxidative species are the lipid radicals, and these are sort of secondary oxygen species. So we have the lipid peroxides, lipid peroxyl radical, lipid oxyl radical, and in general, when these species form, it's your lipids, your membranes that get very brittle. And oxidative stress, it can also affect proteins. Proteins can oxidize and lose biological activity. DNA is also affected. It can become brittle and get strand breaks. So we're in an environment with severe oxidative stress. So here we see the cytotoxicity of reactive oxygen species, and we have the superoxide being metabolized to the hydrogen peroxide, which can then be metabolized to the hydroxyl radical. And we mentioned these reactive oxygen species. They oxidize DNA, lipids, and proteins in a cytotoxic manner. But fortunately, we have some enzymes in our cells to prevent this oxidative stress. One of them against the superoxide is superoxide dismutase. So it protects us against the superoxide. And this enzyme, it's a homodimer that's present in the cytosol. You see the enzymes take up acid, and then they convert the superoxide to water and oxygen and hydrogen peroxide. So we're getting rid of the superoxide, but we're generating then the hydrogen peroxide, which is also bad. So this enzyme has copper and zinc, And the superoxide dismutase, it's a fairly small enzyme, 153 amino acids. It weighs 16 kilodaltons. So the superoxide dismutase is good. It protects us from the superoxide. We also have 3 enzymes protecting us from the hydrogen peroxide. One of these is catalase. And so we have the hydrogen peroxide then going to oxygen and water. So we have a toxic product going to an innocuous, toxic substrate going to an innocuous product. So catalase, it's a tetramer. It's localized to the peroxisome of the cell. And cofactors include a heme group, which means we have iron and NADP. And catalase, it's a larger enzyme, 526 amino acids, molecular weight of around 60 kilodaltons. Another enzyme that protects us against the hydrogen peroxide is the glutathione peroxidase. So we have the glutathione reacting with the hydrogen peroxide to generate oxidized glutathione and water. So glutathione peroxidase, there's several forms of it. The glutathione peroxidase 1, it's a tetramer in the cytosol. It's got a selenium metal that's attached to one of the cysteine amino acids. Glutathione peroxidase 1 is composed of 201 amino acids. It weighs around 22 kilodaltons. The last enzyme that protects us from hydrogen peroxide is the peroxide reductions, and these reduce hydrogen peroxide, organic and fatty acid, and phospholipid hydroperoxides. And basically what happens is a sulfhydryl on the enzyme reacts with an acid group, generating an oxidized cysteine and a water product. So the peroxide reductions, it's a homotetramer, and we have the cysteine at the 46 being oxidized, and then it reacts with a cysteine from another subunit to form a disulfide. So this enzyme has 223 amino acids, weighs about 25 kilodaltons. So we have these enzymes then to protect us from oxidative stress, and that's a good thing. And then the reactive oxygen species, they affect 2 signaling transduction pathways. I'll be talking about this in more detail in a few weeks. But we have the MAP kinase pathway, and in particular, the ERK gets tyrosine phosphorylated. The phosphorylated ERK then goes into the nucleus and interacts with AP1 sites on genes, affecting the proliferation, survival of various cells. A second pathway that's important is the NF-kappa B pathway. This is an inflammation pathway that makes the cells survive. And reactive oxygen species, we mentioned they're important in cancer and in heart disease, as well as stroke. Oxidation, reactive oxygen stress leads to increased aging. You can have alterations of the immune system leading to arthritis. You can get neural degeneration. Once you kill the neurons, they don't come back. They can't regrow. So, if we have all this oxidative stress, how can we shut it down? And the first thing they looked at is vitamins. Fruits and vegetables have a lot of reducing vitamins within them. And studies have focused, especially using vitamin C, ascorbic acid, vitamin E. That's what I took when I lived in Los Angeles. It's an antioxidant, and vitamin A. But these studies with vitamins, especially vitamins in cancer, have been largely unsuccessful, and we'll read this one. The research was carried out in France led by Dr. Serge Kirkberg from the Scientific and Technical Institute for Nutrition and Diet in Paris. It was called the SUVIMAX study. A total of 13,000 men and women aged between 35 and 60 years old were recruited to take part in the study. Half were given daily supplement containing a combination of antioxidants and vitamins, while the other group received placebo. Patients received 6 mg of beta-carotene, 120 mg of vitamin C, 30 mg of vitamin E, 100 micrograms of selenium, and 20 mg of zinc. According to the researchers, the pill's lack of effect may be due to the fact that people in especially Western and European states have a very balanced diet. So as a result, these studies were largely unsuccessful. They found no difference between the patients taking the vitamins and the placebo. But that doesn't mean that antioxidants, if they're used properly, they can be beneficial in certain types of diseases. So we'll see at the end of this lecture, one thing that I've added this year that's new is hydrogen. And so hydrogen is potentially a new way to reduce oxidative stress without causing cellular damage. So nitric oxide, this is Dave Wink's specialty, and we have nitric oxide synthase enzymes catalyzing the reaction using arginine as a substrate to citrulline and nitric oxide as a product. And one important cofactor is oxygen and NADPH. So we have 3 types of nitric oxide synthase. We have the neuronal, the inducible, this is big in cancer cells, and then in endothelial cells, they have the enos. And here we see the reaction. We have arginine as a substrate. We have the oxygen being metabolized to water using the NADPH. Then we attach oxygen to the nitrogen, and then in turn we end up with nitric oxide as a product as well as citrulline. And hydroxy L-arginine is an intermediate in the reaction. So here's our reactive nitrogen species then. And the instigator is nitric oxide. And then nitrogen dioxide, this is a pollutant that comes from engines burning gasolines. And so in Los Angeles, often the sky was very brown at midday, and that's from the nitrogen dioxide. And then other important factors include the nitrite. This is used to cure meat. And so as a result, the meat bacterial can't survive in this. Another thing it does is it turns the meat pink in color. So oftentimes you'll go to butchers, early in the morning you'll see they're working with brown meat, and then half an hour later it'll be pink, and that's because of the nitrite that they've added. And then we have nitrate as well. This is present in a lot of vegetables. And under certain conditions, nitrite and nitrate can produce nitric oxide. And then a very bad factor is the peroxynitrite. This is very toxic. And then Dave will be talking later today about S-nitrosyl thiols and nitrosyl metal complexes. So in terms of the biology, an important factor is hypoxia. And so we have this enzyme, prolyl hydroxylase, and it's inhibited by hypoxia as well as reactive nitrogen species. But when this enzyme's active, then it will cause hydroxylation of this factor, HIF1 alpha, and then it will bind to von Hippel-Lindau protein, get ubiquitinated, and degraded. That's shown in the next slide. We see here's the prolohydroxylase. And when it's active then, as shown in green, the HIF-1 alpha gets oxidized, von Hippel-Lindau protein binds to it, gets ubiquitinated, and degraded. But another factor that gets degraded is p53, and it can also get degraded. But under conditions of hypoxia, as well as reactive nitrogen species, the p53 gets phosphorylated, altering gene transcription. And similarly, the HIF-1 alpha can then affect the gene activation. So HIF-1 alpha, as it accumulates then, it can form a heterodimer with HIF-1 beta in the nucleus of cells, and the P300 CBP then binds to the HIF-1 alpha complex, altering gene expression. So this is the biology then of reactive nitrogen species and hypoxia. So what does HIF-1 alpha do? It helps normal tissues and tumors survive hypoxic conditions. It's a transcription factor which alters the expression of 40 genes. And the group 1 genes include transferrin and VEGF. And this causes cells then to deliver more oxygen to tissues. Group 2 genes include enolase and hexo-1 kinase, which provide sugars to cells to generate energy. Group 3 genes include IGF-2 and IGF-1 binding proteins 1 and 3, increasing the proliferation and viability. So HIF-1 alpha then, it basically protects the cells and keeps them going. And it's really important when the oxygen concentration is less than 4%. So with hypoxia then, we see in green, we get more HIF-1 alpha, altering the gene expression. We get more of the phosphorylated p53, altering the gene expression. So under normal oxygen conditions, p53 can bind to MDM2 and be degraded. Reactive nitrogen species downregulate MDM2, resulting in less p53 degradation. And then we get phosphorylation of the p53 at serine 15, and then this alters the gene expression through the P300 CBP. So then, under conditions of low oxygen or under conditions of reactive nitrogen species, we then alter the gene expression. And in particularly, we generate more energy in the cells through upregulation of hexokinase 1. We stimulate angiogenesis, resulting in delivering more oxygen to the tissues through VEGF. And we stimulate cell proliferation through the IGF-2. So in cancer then, the big thing is the carcinogenesis process. So carcinogens lead to DNA damage, and if you do things like smoke cigarettes, initially you just get benzapyrene, which doesn't do much, but then it gets oxidized by oxidative stress into carcinogens, and that starts the carcinogenic process. So initially in colon cancer cells, you turn on APC and beta-catenin in early events. In secondary events, then you turn on various growth factors. So you get an initial carcinoma, you stimulate the production of cancer cells, and then with further mutations, such as mutation of P53, then you get a malignant tumor. And the tumor can then undergo metastasis from the primary organ to distant sites, such as for lung cancer, my specialty, you go from the lung to the lymph node, liver, bone, and then ultimately the brain. So cancer cells then, what happens is these small tumors have plenty of oxygen, but as the tumors get big, then you need angiogenesis to deliver oxygen to the tumors. So when the tumors get bigger than 1 millimeter in size, the host delivers blood vessels to the tumor containing oxygen and nutrients required for further tumor growth. Then once the tumor grows, it can escape the primary organ, get into the blood or the lymph nodes, and go to a secondary organ. And so for lung cancer, one of the first places it goes is the liver, and you can look at a patient's liver and you'll find very little functioning liver cells. The tumor has taken over the liver. So this can lead then to liver failure and death. So here we see an example of a tumor, and as we get further away from the blood vessel, there's going to be less and less oxygen present. The oxygen is going to be delivered from the blood vessel to the tumor. So initially there's going to be high concentrations of oxygen, but then that's going to go down with distance. So here you see a chart just showing the oxygen levels, and in breast cancer you see right next to the blood vessel you have a fairly high concentration of oxygen, almost 10%, but then as you get further and further away it goes down to less than 2%. So in terms of hypoxia then, the transcriptional activity of the AP1 sites is increased, leading to nuclear oncogene production, such as CPOS and C2. And NF-kappa B, it's strongly activated when cells are re-exposed to normal oxygen. So initially, with hypoxia and tumors, the AP1 sites are turned on. And then when the tumor is re-oxygenated, the NF-kappa B can be then turned on. And for tumors, the problem is if they don't have enough oxygen, then you can give radiation therapy, and the overall survival of the patients isn't very good if the tumors are very hypoxic. But if you can re-oxygenate the tumors before radiation, then the patient's survival is increased. So this is what's called a survival curve. It's done at the clinical center all the time. For NCI over in Building 10, we have over 200 clinical protocols going at any one time. So now we're going to switch gears to hydrogen sulfide. So again, this is a gas. And hydrogen sulfide, it inhibits caspase cleavage and also p38 MAP kinase phosphorylation. So then this would lead to increased survival of cancer cells. So hydrogen sulfide then, it's primarily obtained in cells from amino acids, and we have 2 amino acids with sulfur, the methionine and the cysteine. And it turns out the methionine is an essential amino acid in your diet, and the cysteine is not. You can get cysteine from methionine through metabolism. So we see we have a whole series of enzymes. Methionine synthase converts methionine to homocysteine. Cystathionine beta synthase converts homocysteine to cystathionine. Gamma cystathionase converts cystathionine to cysteine. And then the cysteine can be further metabolized to mercaptopyruvate and then ultimately pyruvate. We see hydrogen sulfide can be released here. It can be released here. And you know when it's released. Hydrogen sulfide, it's the smell of rotten eggs. Or in my case, I grew up in California. You go down to the bay, and the bacteria there will produce hydrogen sulfide, so it's stinky. Another place it's produced is when you have seismic activity, such as earthquakes. So, it was found then that if we eliminate one of the enzymes and knock out mice, they have reduced hydrogen sulfide, and this leads to heart disease and hypertension. And loss of a secondary enzyme in humans leads to homocystinuria, which affects the cardiovascular system, the skeletal system, and the ocular system, as well as the CNS. So in humans, these enzymes can be genetically lost through diseases, and about 1 in 100,000 people are homozygous recessive for these particular enzymes, leading to disease. So hydrogen sulfide, basically, we find at low concentrations, stimulates oxygen consumption, but at high concentrations, it can be toxic. It can inhibit the mitochondrial respiratory chain, as well as cytochrome oxidase. So glutathione is sort of a cellular protective agent. It's composed of 3 amino acids, glutamate, cysteine, and glycine. And it's made, we have 2 enzymes that are important, the glutamyl cysteine ligase, the glutathione synthase. And so glutathione, it helps keep things reduced. And cancer cells, if they're exposed to severe oxidative stress, they'll make lots of glutathione, as much as they possibly can, because then that helps them survive. So it's an antioxidant, and it provides cellular cytoprotection. It maintains the redox environment of the cells, and it's increased when cells are stressed and downregulated after a challenge has been met. And so here is real cellular biology. We're now exposing a cell to oxidative stress, and we see that superoxide is made, and then it can be metabolized by superoxide dismutase to hydrogen peroxide. And then the glutathione peroxidase is going to reduce the hydrogen peroxide. And in the process, the glutathione is going to get oxidized. And then what the cell is going to do is it's going to reverse the glutathione and reduce it through glutathione reductase. And then we're going to get glutathione again, and it can meet the next challenge, the next stress of hydrogen peroxide. So this is really how cells work. We have these enzymes, they're working together, and they're regulating the oxidative stress. So another gas that we want to talk about is carbon monoxide. And carbon monoxide can be quite toxic. And what happens is it binds with very high affinity to the iron in hemoglobin, higher affinity than does the oxygen. So if people want to commit suicide, one of the ways they can do this is to turn on their car engine in a garage, and then the carbon monoxide gas will come out from the engine, and it will bind to your hemoglobin, and you'll die from suffocation. So if you want to save someone who's been exposed to lots of carbon monoxide, you just got to take them out and give them lots of air. So once the CO is released, the hemoglobin is also degraded. Carbon monoxide also inhibits cytochrome C oxidase, but it maintains the cellular ATP levels, resulting in the generation of reactive oxygen species. So carbon monoxide addition to cells results in P38 MAP kinase phosphorylation. So CO levels are reduced by the heme oxygenase. And so the heme oxygenase, then it gets the carbon monoxide released from the heme, and the patient will slowly recover. And the last thing we're going to talk about is hydrogen gas. I got back a week ago from a meeting in Japan, and this is one of the things that they were talking about. And I found it very peculiar because in the airports of the US, I noticed they were selling water in bottles, and it said it was oxygenated water. And then I get to Japan, and they're talking about hydrogen water. So the Americans are very interested in oxidation. The Japanese want to get rid of the oxidative stress, and so they can use hydrogen. And so basically, the hydrogen, it's a gas, and it functions as an antioxidant. It gets rid of hydroxyl radical and peroxynitrite, two very toxic things. And the Japanese have found, especially in animal models, it's anti-inflammatory, anti-apoptotic, anti-allergic, and it stimulates energy metabolism. So it sounds real good. Solves a lot of our problems. And so basically, it's important in the cancer field. The reason is, we mentioned before, to get carcinogens that lead to DNA alterations, the chemical has to be oxidized. But we'll also see that it's important in diabetes, and we have an example of neurodegenerative disease as well. So the interesting thing about hydrogen is it readily crosses the plasma membrane. It goes right through the plasma membrane, and it can accumulate in the cytosol or various organelles, such as the nucleus or the mitochondria. And it doesn't really affect redox homeostasis, but if you have excessive oxygen, reactive oxygen species, that's what it goes after. And so initially, they were trying to give gas to the animals, and that didn't work so well. They found a better way was just to put it in the water. And so using gas, they could get up to 10 micromolar hydrogen in the blood of the animals. But using the water, they could get 80 micromolar. So they could get much more hydrogen into the blood using H2-infused water. But the trick is it has to be stored in an aluminum container. If you put it in plastic or glass, it just doesn't work. The hydrogen just goes away. So in this way, using the aluminum container, they can just infuse the hydrogen in the water once a day and give it to the animals. And then chronic oxidative stress, they studied it in a Parkinson's disease model. So in Parkinson's disease, you have neuronal loss in the substantia nigra. And they found that if they gave the hydrogen water to rats after treatment with 6-hydroxydopamine, or MTT, that there was less loss of dopamine neurons, and it improved the mental state of the animals. So basically, what they're saying is when you give the 6-hydroxydopamine or the MTT, this causes lots of oxidative stress, resulting in neuronal loss. And if the animals are loaded up with hydrogen in their blood and cells, then this will reduce the oxidative stress, and you'll have less loss. The loss of the neurons will be not as great. So a second animal model that they looked at was the diabetic mouse with the fatty liver. So this is an animal model for type 2 diabetes, and they found the hydrogen water did not affect the food intake or the water consumption of the animals, but it decreased the fat content and the body weight. And they also found then that the plasma glucose decreased, the insulin and triglycerides, and it basically stimulated energy metabolism. So this potentially is something that's going to be very important in the future, using hydrogen as a therapeutic agent. So in summary then, we've sort of gone through reactive oxygen species, reactive nitrogen species, we've talked about hydrogen sulfide, carbon monoxide, and now a new area that might play a role in therapy is using the hydrogen gas. And so we have some references then for the hydrogen sulfide and a brand new reference for the hydrogen in medicine. And that will conclude my lecture. Are there any questions? If not, we're going to bring Dr. Wink up here. He's got a lot to talk about. He's one of the world's experts in nitric oxide. And so as an introduction, he got his degree at the University of California, Santa Barbara. And surfing, no, they don't surf there. That's where they have all the oil wells in the water of the ocean. So the surfers don't like to get oil on them. But after being at Santa Barbara, he then went to the Massachusetts Institute of Technology. He joined NCI at Frederick. And then he came down here to Bethesda at the radiation biology branch. And now he's back in Frederick with the cancer inflammation program. And he's organized several meetings, such as the Gordon Research Conference in Barga, Italy. And he's also a fellow of the American Association of the Advancement of Cancer. So let's see, here we are, Anthony. He's going to put on Dave's PowerPoint, and off we go.
Well, Terry, thank you very much. And the next two lectures are mine. Since we have this redox biology faculty, I thought we'd come in, try to teach people about some of the aspects of it. So I got the hard part. They said, you're the chemist, you have to teach the chemistry. So if you think this is like freshman chemistry in the morning, I apologize. But the idea here is what I want to do in the next two lectures is kind of give you a working vocabulary of the chemistry of redox biology. So one thing about redox biology is, Terry, thank you very much for that lecture, Terry. He shows you a kind of a smorgasbord of what we're going to be talking about over the next several weeks. And the chemistry, one thing about the redox biology, which is a little different than a lot of the biology and molecular mechanisms that we talk about, is that the chemistry actually matters. The actual reaction of these molecules matter and dictate the biological outcome. It's not just simply fitting into a lock and key type theory. In fact, these chemical reactions actually can be modified in different ways to actually create what we like to call a redox landscape. So what we want to do today here is just kind of talk about some of the basic chemical reactions, definitions, and if you have any questions, please ask. It's a lot easier to ask them, instead of waiting till the end and all that. So anyways, So let's talk about the introduction to the redox molecules. Terry's already touched on this. There are several categories here. Now remember, these are small molecules. These are small reactive molecules. They're components of the smog. I like to think of them as some of the building blocks in the primordial soup that gave us life, right? You have hydrogen gas, you have oxygen. Remember, why are we even concerned with redox biology? Because we breathe. We actually utilize oxygen. And that's why we have redox biology. So nature over several billion years has learned how to use these chemical reactions here to actually control many biological mechanisms. So reactive oxidant species, and here, good. AB was not one of my big suits. I actually should have taken an AB course. I go to places all over the world. I never know how to work the machine. So anyways, we have reactive oxygen species, hydrogen peroxide, superoxide. These are the primary ones, and they give host to these reactive species here, which can be produced in things like radiation. Oh, sorry. And then we have the nitrogen oxide species. And I separate these because often these two, the reactive oxygen species and the nitrogen oxide species, they actually often antagonize each other in the signaling mechanisms. But one of the common things, nitric oxide, which has emerged over the last 20 years as one of the most important molecules in the body. I have to throw that because that's what I'm actually in. And they gave a Nobel Prize out for this, of nitric oxide and the control of the cardiovascular system. But in an aerobic environment, NO, as Terry mentioned, can make NO2, N2O3, and in the presence of superoxide, make peroxynitrite. These are actually very reactive compounds, and they can modify different chemical stresses. HNO is one that we actually have been interested in for many years. This is a one-electron product. We call it nitroxyl. And the interesting thing about nitroxyl is that it actually has orthogonal signaling in biological systems from nitric oxide, even though it's only reduced by one electron. In fact my collaborators at Johns Hopkins has actually shown that HNO is critical in increasing the inotropic and chronotropic effect, inotropic and lusotropic effects of the heart. In other words, for heart failure, it actually increases the muscular work. And there's actually a drug that was developed based on this HNO technology that's now in phase 2 clinical trials. So just by changing one electron here, you can get vastly different things. And you can actually get interesting biological effects. Carbon monoxide, of course, CO. Everybody knows about it. It binds only to ferrous compounds. Unlike the reactive nitrogen oxide species or the reactive oxygen species, CO pretty much does one thing, and one thing only. Now, I might be surprised some other, later on, somebody may come out with something unique, but CO pretty much binds to metals, and that's it. So when we talk about different types of classification of these chemical species, instigators, we have primary reactive species that are usually conversion of these species here, which are fairly stable, into reactive species here. And then some of these reactive species react with biological molecules, form secondary species. You often get a biochemical product and ultimately a biological effect. So the best representation here of the oxygen is peroxide reacting with metals to form either these iron oxo species or hydroxyl radicals. These can react with things like lipids or proteins to form lipid radicals or carbonate radicals. And these can go on to oxidize different types of proteins like different type of macromolecules such as DNA or further oxidize lipids. And ultimately, this from hydrogen peroxide will induce what we like to call an oxidative stress. So on the other side, we have nitric oxide, and nitric oxide is a simple molecule. And actually, you can buy a tank of it, so it's fairly stable. And at low concentrations, I don't know if you're familiar with nitric oxide, but it was given the Nobel Prize for its discovery of the endothelial-derived relaxation factor. This small molecule here at low concentrations controls blood pressure. It actually causes hypotension. And it's a critical regulatory mechanism in the endothelium in controlling your blood pressure. If you have high blood pressure, they give you things like nitroglycerin, which donate nitric oxide. So NO, though, in the presence of, at much higher concentrations, can lead to NO2, These are the primary reactive species. And these can form a host of different types of modifications of our biochemical products, which can induce what will, at high enough concentration, induces what we call nitrosative stress. So, what is the concept of redox stress? Now, this is something it's been thought about we'll call it discussed. Often people yelled. One guy actually had a heart attack during one of these things at a meeting, but that was 20 years ago. And so, but at redox chemistry here, even if you do produce a reactive molecule, that does not necessarily mean it's bad. It's just reactive. So a lot of people say, oh, I've generated the hydroxyl radical, or I've generated these oxidized lipids or nitrolipids. Well, biology has an interesting way of compensating for this. So you have to have this biological balance here, redox balance here, where you have a chemical stress, where you have so much of it that it overwhelms all the systems, all the protective systems in the biological system. However, these redox chemicals are actually really, really important in controlling homeostasis or biological homeostasis. Controlling vessel tension is very, very important, and is actually controlled by ROS and NO. These are the major players that actually control the contraction or relaxation of the blood vessels. And anytime you get an imbalance of these, it can result in disease. So I like to think of stress as more like an atomic bomb, or we can take nuclear power or whatever, and you can actually use this beneficial wiring inside a building here. So it's actually has to be taken in context. And how we determine this context is actually trying to understand the chemistry. So what are the chemical effects? Well, the direct mechanisms of the chemistry of oxidative stress. We're going to talk about oxidative stress because, I mean, in the public lexicon, right, we have if you go and get your shampoo, it has antioxidants in it, right? So vegetables are good for us because they have antioxidants. So this is part of the lexicon. So let's try to understand at least the chemical and even biochemical basis of oxidative stress. So the chemical effects here are where the biochemical reactions directly destroy cellular and tissue structure. Lipid peroxidation, which we'll discuss next week in some detail. Metal catalyzed oxidation of DNA. You throw hydrogen peroxide. Has anybody thrown hydrogen peroxide on your cells? You throw enough of it on, it dies, right? But if you put a little bit on, you can actually cause growth. So the chemical effects and understanding the targets of these is very important. Production of oxidized products. Well, oxidation doesn't only have to kill, we can get oxidized cholesterol, and that's not good. Some of these oxidized lipids are not very good. DNA base oxidation and misrepair can result, as Terry talked about earlier, some genotoxic events. So changes in the biological redox balance, and this is really important. This is where I like to define where people used to think about. I remember one guy was being interviewed on one of those talk shows. I think it was Dick Cavett. This is before you guys were all born. And so not him, but everybody else, right? And so guy got in there and says, we're being irradiated all the time because we're producing these hydroxyl radicals, which radiation hits water and it produces hydroxyl radicals. So therefore, just by the virtue of living, we are being irradiated all the time. Well, that's not really quite true. Mother Nature is much smarter than that and has had 1 billion years to figure this out. But one of these things is the ischemia reperfusion injury. If you block off blood flow to a tissue, there's sensors that sense that there's no flow. And then there's no oxygen. So when the flow comes back, all this hypoxia now has all this reducing power, and it pummels those electrons to produce hydrogen peroxide rather than just simply burning it. And a lot of this ends up recruiting a lot of leukocytes because a lot of the oxidative stress on the venular side. And if you put in a little bit of NO, you actually ameliorate this leukocyte adhesion here, and you can actually dramatically reduce things like stroke. Shear stress in the coronary circulation is another one. So there's almost every process, I think, in your body has some sort of balance of redox that's involved. So the chemistry outline, what we'll talk about today is some of the fundamental concepts. Redox biology, you're actually transferring something. Either you're transferring an atom of oxygen, a hydride atom, a hydrogen atom, or electrons. And you're actually causing a valence change. Chemistry formation of these reactive species that you'll read about. And some of these, I want to familiarize you with the DNA, the proteins, the lipids. And then finally, I want to talk a little bit about the chemistry between ROS and NO, because I think over the last 20 years, we've realized that this is a balancing act and is very important in really understanding biological effects of redox biology. So this, I promise you, since even though I was a physical chemist by training, I don't admit that often, but I was. We did teach this, electron transfer. This is the famous Nernst equation here. And basically it says that if you have oxidized or reduced, the electron transfer, if it has a positive E value here, then that half potential is favorable. If it has a negative, it's unfavorable. And if you produce this, it wants to get rid of its electrons. So that probably saved you about 4 lectures in freshman chemistry if you just knew that, right? So, for instance here, okay, so one of the ways that if you want to go in and detect superoxide here, and a lot of people do this, is the classic ferrocytochrome C. Now, ferrocytochrome C is an iron 3 complex, which is pretty much hexacoordinate, although there are some pentacoordinate, but mostly hexacoordinate. So basically, it takes an electron out, and it can accept the electron. And when you put an electron in the ferrocytochrome C, its potential is plus 0.26. Now, superoxide, when it's produced, actually is more unstable than oxygen. So what that means is that when it gives up an electron, it's actually, this reaction is favored by 0.33, and when we add them together, it's 0.59. So that means this reaction is favorable. And in fact is that if you really wanted to, these type of reactions, you can go back through here and actually calculate out an estimated K value, or a rate constant for this. So this is a fairly facile reaction because it goes downhill. So if we start looking at the pecking order of oxidants, and this I get this from Gary Buettner, I think published this from Iowa, who is the other people who put on the course like this. This potential up here means a hydroxyl radical here, which always is the granddaddy of all oxidants here. It's produced things by ionizing radiation, by splitting water. Many chemical reactions can actually produce this. And the hydroxyl radical has a 2.3 volts. So this is, it really wants electrons here. These are lipid radicals, and you can see they're less, but they're still pretty powerful. I'd actually say anything above ascorbic acid here is really a powerful, or is a pretty good oxidant here. And things like tocopherol is a kind of a mild oxidant. But boy, I'll tell you, anything over a volt or above is a pretty powerful oxidant and can be harmful. If we look down on the other side here, you can see like paraquat. Well, paraquat, to reduce paraquat, it takes a lot of energy here. To reduce oxygen, if you reduce iron to iron 2, that's actually fairly mild. So by using this, you can actually start seeing and start ranking reductants and oxidants. So what is redox chemistry and what is electron transfer? There are 2 types of electron transfer. Is there any questions so far? So, and this is really important. This is often skipped by a lot of people. And the electron transfer here, the oxidation of, if we look at oxidation of substrate, we get the electron, we take an electron out, reduction, we put it in. But one of the most important concepts that's overlooked is outer sphere versus inner sphere electron transfer. And they actually gave a couple Nobel Prizes out for this, Marcus and Taube. And so outer sphere electron transfer is basically where the electron just skips without making a bond, skips from one atom to the other. It's sort of like the static electricity. On the other side, we have inner sphere electron transfer, where you actually have to form a bond. And that bond, actually the electron transfers. And so here, this is just basically how molecules collide, but this is dependent on whether or not you can make a bond. And one of the big differences here is the reduction of oxygen by flavoproteins. The flavoprotein can be reduced. It can donate electron directly to oxygen. But thiols, we often think about thiols as redox active, and they are. But they actually do this through making bonds, sulfide bonds. So that's a big difference in sulfur chemistry versus just simple electron transfer. And one thing I've learned through 20 years of redox chemistry, just studying this in biological systems, Mother Nature does not like to do outer sphere electron transfer. You don't want to put an electron just having it skip around because you have the potential in the presence of oxygen and other things of making powerful oxidants. So let's talk about outer sphere electron transfer, some of the classic ones. We have reduction of oxygen. Oxygen can be reduced by one electron to form superoxide here. There's nothing really super about superoxide. It just sounded cool, I guess. But in the case of superoxide here, superoxide's actually fairly unstable. And either in the presence of the catalase, superoxide dismutase, or just spontaneously, it dismutates. In other words, it forms hydrogen peroxide and water. So even if you produce, a lot of people think that if I produce superoxide, it just sits around. Yes, you can buy superoxide, by the way, potassium superoxide, but it is fairly unstable. When you put it in water, it actually explodes. It's kind of fun to have the people in the lab do that. It foams a lot. But in this case here, oxygen can also be directly reduced by 2 electrons to give hydrogen peroxide here. So this is the basic, fundamental chemistry of the interaction between oxygen superoxide and hydrogen peroxide. Now, hydrogen peroxide, if you put 1 electron in, you produce this hydroxyl radical here, and H2O. And this is what they classically call the Fenton type reaction. Metals donate an electron to hydrogen peroxide and generate this disproportionation to water and hydroxyl radical. The catalytic activity, or what catalase does, it takes peroxide, and actually instead of producing hydroxyl radical, it makes water and oxygen. And this is actually a very important detoxification pathway in vivo. Or, in the case of respiration, or some other process, you can put 2 electrons in and make 2 waters. So H2O2, no matter what, it's actually fairly stable because you can buy a bottle of it. But it's actually, you require a metal catalase. And that generally is iron and copper, though other metals can do it. In biological systems, it tends to be iron and copper. So here's some examples of intersphere electron transfer. And in this case here, we have a disulfide bond. And if anybody's tried to stabilize proteins with mercaptoethanol or try to reduce it with DTT, basically what happens is that you take reduced DTT and you put it in, you oxidize the DTT to form this disulfide bond here, and then you actually can reduce your protein. Lipoic acid is another natural way of reducing disulfide bonds, and so is thioredoxins, which is actually a critical protein to actually maintain the sulfhydryl population. Hydride transfer is another one. Hydride transfer is classic. It's almost in most every system that undergoes some redox with NADPH or NADH. NADPH can donate a hydride to FADH to reduce FAD by 2 electrons. Now, hydride transfer here is a great way of transferring electrons without the intermediacy of a free electron. So, oxidation mechanisms of biological molecules. Again, let's talk about the hydroxyl radical because you'll actually read about this more than anything. I think the hydroxyl radical is produced in vivo, but also metal oxo species, which we'll talk about when we talk a little more about the Fenton chemistry, can produce equally oxidizing species. Here's the hydroxyl radical. But in the case of the hydroxyl radical, you can oxidize things, almost anything, to a radical species here and water. Ribose, for instance, could be oxidized to form a ribose radical here. And the hydroxyl radical formation of things like these guanine radicals here can actually undergo various different rearrangements here to produce different products. And these products here are important because they can be detected as oxidized nucleotides and determine under a biological condition whether or not there's oxidative stress. There's also what I like to call oxygen insertion reaction additions. And in case here, we'll talk about this a little later here, but these metal oxo species, which can be produced between metal and hydrogen peroxide, can react with a nucleophile and insert an oxygen here. We'll talk a little bit about methionine later. In the case here of methionine type molecules here, you can see we can oxidize here the methionine to a sulfoxide and an iron complex. And that is actually fairly important because many of the products that we do see when we're detecting some of these, even in the gas phase, come from this type of reaction. So what are the special chemical terms of nitrogen? Is there any questions so far? Oh, go ahead. We're gonna get to that. But the big difference there is that in the heme, because it's what they call softer, because you have all these pi electrons, you actually produce the iron oxo species there. And that stabilizes that species, and also in a pocket that allows another hydrogen peroxide to come in there and form water and oxygen. In the Fenton chemistry, you don't have this big, often you don't have this big biological organized complex around it. And that chemistry can happen, but it happens more rapidly. Other substrates come in, and that iron oxo species there can oxidize different substrates. Now, peroxidases can do that if you set up the conditions right. But in the case of catalase, because of the shape of the pocket region, you can actually control that chemistry. So it's a good question because the chemistry is a little bit different, but it's more about the shape, where and when that makes a big difference. I think next week we'll talk a lot more about the peroxidases and catalases. So here we have different terminologies here for nitric oxide that you probably read in the literature. Nitrosylation, this is actually a term that we've been talking about a lot, but from a chemical perspective, nitrosylations really just simply means the addition of NO to a substrate to form a nitrosyl group. Metals are classic. Metal plus NO makes a metal nitrosyl. Radicals, which are very reactive radicals, they can react with nitric oxide because, I don't know if I said this, but nitric oxide actually has an unpaired electron. So it too is a radical, but it's a stable radical, and it can make nitrosyl complexes. Nitrisation, which actually a lot of the chemistry proceeds by, is actually the addition of NO+. Now, NO+ itself doesn't really exist in biological systems. They actually are attached to things like nitrite or chloride or other things, and they donate an NO+ to modify the nucleophile. So it's a nucleophile plus an electrophile actually give you this RNO. And one of the classic cases here is N2O3, which is formed from the NO-oxygen reaction and other chemical reactions involved with nitric oxide. It binds to thiols and forms an S-nitrosothiol. The fact is, over the last 10 years, S-nitrosothiol has actually emerged as a potential signaling pathway that's alternative to a lot of just simply ligand receptor types signaling. Amines plus N2O3 can produce nitrosamines. Now, nitrosamines back in the 1970s was a carcinogen. Well, it still is a carcinogen, but they were discovered to be carcinogens. And one of the classic stories about nitrosamines was, I think in the 1970s, they discovered nitrosamines were being produced in German beer. Well, this caused, of course, a national crisis in Germany because they had to shut down the beer industry. But within 6 months, they actually had solved it and found out that nitrosamines were actually being produced in the processing of the hops, and they were able to change that. And still, nitrosamines tend to be a little more important, although it seems to be nitrosamine carcinogenesis, very carcinogenic, some of these nitrosamines. However, nitrosamines in humans actually may have less effect than they did in rats, but we can talk about that later. Nitration, you have nucleophiles of NO2+, and the classic one is tyrosine and nitrogen dioxide here forming nitrotyrosine. And nitrotyrosine can be a footprint you actually have an antibody that can measure this, is a footprint for the presence of nitrosative stress or reactive nitrogen species. So the general chemical reaction here is the Fenton reactions that generate these powerful radicals is iron 2 plus hydrogen peroxide can give the hydroxyl radical ferric ion, or it can actually generate this iron oxo species here. In fact, there's been kind of a debate over the last 30 or 40 years of whether it's really a hydroxyl radical or whether it's a metallo-oxo species. But one of the things about whether you get a species that is more reactive, like iron oxo species, or more or less metal oxo species, goes back to your question here. What is the difference between heme? I mean, same basic chemistry, right? But it's the difference here is these hard ligands as well. So if you have a lot of hard ligands like oxygen and nitrogens around a metal center, they will actually tend to favor more of a reactive hydroxyl radical type character. If you have a porphyrin or sulfides around it, in fact, you think of these softer ligands as buffers for this oxidative stress. It stabilizes. When we mean buffers, orbital buffers, it means like a stabilizing factor. And in fact, in that case, they're a little bit less reactive, though they are pretty reactive. I mean, P450s are fairly reactive. They do some impressive types of chemistry. But again, it comes to location of where this happens. And also, we can tune these metals here to generate different types of chemistry, which is kind of neat. Heme complexes here. I don't know if I actually think I got this out of a book or Wikipedia, something like this. But anyways, here, this is just your classic heme. And these are flat porphyrins. They have this delocalized pi orbital here, which actually interacts with this metal here. And the redox chemistry, these redox molecules, are very much also affected by the distal ligand here. And many of the heme proteins have histidine. These would be like hemoglobin, myoglobin, the peroxidases, or cysteine like cytochrome P450, nitric oxide synthase, or tyrosine like catalase. And these ligands here control, this goes back to differences between Fenton chemistry or even different metal proteins. Their chemistry is dictated, even if they have the same porphyrin ring, by what this X group is here. Because in the case of things like cysteine can actually also interact with the metal through these pi systems as well and change the type of chemistry that it's doing. So peroxidase chemistry, in this case here, we have most peroxidases are in this iron 3. Iron generally is an iron 2 or iron 3, even though there are other valence states it can be in, like iron 4 or even iron 5. Those are very powerful oxidants. But the resting states tend to be 2 or 3. Iron 3 plus peroxide can give you these, what they call pication radicals. So if you actually go in the lab, you have peroxidases, and you do an assay, one of these colorimetric assays. You put in hydrogen peroxide as a developing agent. It's usually peroxide in there, which is reacting. And this is the type of chemistry. Then this oxidizes substrates, usually some sort of dye. Excuse me. And you get, this is compound 1, which is this pication radical. You reduce it by this compound 2, and it goes back to the native state. This native state can be reduced here, and in some cases, and a substrate, then this can react with peroxide, and then you can actually form this loop here. But basically what happens is hydrogen peroxide reacts with this, give this pication radical, and this then oxidizes substrate back to the native state, and you form a catalytic mechanism. Things like myeloperoxidase will actually oxidize chlorides, and that's one of the best examples. You're producing chloride, right, or hypochloride, HOCl, which you actually put in your pool. So if you have chlorine wipes and things, that's what you're doing. And your body actually has an enzyme that's producing this, which they call myeloperoxidase. Nitrite, which has emerged here, can also be oxidized and produce a powerful oxidant, NO2. And why does the body want to produce NO2? Well, NO2 is very good at fighting some types of pathogens. And things like tuberculosis. It's very sensitive to NO2. So the ability for these types of enzymes to oxidize nitrite, which is actually in your diet, spinach, nitrates and nitrites, and they're interconverted through your microflora, actually, in your oral cavity and your gut cavity. Nitrite can actually produce NO2, which can be a powerful oxidant to fight different types of pathogens. Superoxide. Well, superoxide can give up one electron. This is kind of a unique molecule here. Superoxide can produce oxygen by giving up one electron, as we discussed earlier. But superoxide can also take an electron and produce hydrogen peroxide. And this latter reaction here is a classic Haber-Bosch reaction, where you take iron 2 plus hydrogen peroxide, you generate a hydroxyl radical or an iron oxo species. This species can oxidize hydrogen peroxide, taking one electron away, going back to superoxide. But superoxide, interestingly enough, can reduce iron 3 back to iron 2. This comes back to here, and you form this cycle here, which you can actually, under these conditions here, you can actually catalytically generate powerful oxidants like hydroxyl radical. I want to tell you this one thing because I should have put this down here. But when you're working with redox chemistry, there's things called data pack, or these iron chelators. What do these iron chelators do? These iron chelators prevent this Haber-Bosch chemistry from converting iron 3 to iron 2, and actually can prevent any just stray or peroxide reaction. We actually use this a lot in many of our different assays. So the dismutation of superoxide here, we have, again, it's pH dependent, as Terry mentioned, superoxide dismutase. Now, there are really three forms of it. There's the copper zinc, which is in mammalian cells that's in the cytoplasm. There's the manganese, which is SOD2, which is in the mitochondria. And you have an iron bacteria. And this, a lot of groups here, like Erwin Friedovich's group, have described these over a number of different years. And what happens here is that in the case of the copper, is superoxide reduces copper, waits for another superoxide, and then the electrons donate to superoxide to form H2O2. And there's a lot of people who SOD is protective in a lot of different mechanisms, probably because it's not allowing electron transfer to other metals, and like in the Haber-Bosch reaction here. And there are a lot of different types of compounds here. Jim Mitchell, who will talk later about later on this fall, talk about SOD mimetics like nitroxides, and there are a lot of manganese compounds. And one of the things that controlling superoxide does, it prevents the inactivation of a conatase. The Haber-Bosch cycling is prevented, and it also prevents the scavenging of NO, because superoxide actually is a very good way of controlling nitric oxide bioavailability. And to give you an example why that's important is in things like atherosclerosis. So if you have your level of NO is keeping your vascular tone, but as you increase oxidative stress, it scavenges the NO. And the scavenging of NO causes vasoconstriction, and it also causes leukocytes to adhere to the vessels. So the reactivity of hydroxyl radical here, in general, hydroxyl radical, one thing about hydroxyl radical is everything is downhill from there. So it reacts with things like carbonate. You don't normally think of carbonate as being reactive, but if actually hydroxyl radical can form carbonate radical, and this is actually a pretty powerful oxidant, not a super powerful oxidant, but it is, making NO2, which is about a volt, so it oxidizes a lot of different things. This is, again, nitrogen dioxide. And hydroxyl radical create organic free radicals. There are a lot of ways how you detect this. Is one of the classic ways is through spin trapping, of using electron spin resonance. And in this case here, hydroxyl radical plus DMPO here can form a stable radical. In fact, Ron Mason's group has devised a really interesting system that using DMPO, he can actually look on things like free radicals formed on DNA and other molecules here. He's developed an antibody to it. So he reacts, so under a certain condition, he can actually trap these radicals here, these free radicals out on macromolecules, and he can put an antibody on there to find out where the reactions are. Other ways of trapping and detecting are benzoic acid, salicylic acid, HPLC is a great way to detect a lot of these systems and work with this. And defendant-derived oxidants in DNA chemistry. One of the things is oxidation of ribose part ends up with strand breaks, DNA strand breaks. This is generally single-stranded breaks. Radiation and other compounds can generate double-stranded breaks, but in this case, single-stranded breaks is generally what defendant chemistry does. You can also oxidize base pairs, induce cross-reaction, and induce deamination. I only throw this up here. That's in your folder for a reference point. I don't expect, it won't be on a test or anything like that you actually have to understand this chemistry. But basically what happens here is if you oxidize into different, like guanine can be oxidized to 8-hydroxyguanine here. And the mutations that result from this type of modification are what we like to call transversions, which is a GC to TA transversion type of mutation here. This compound here can actually go on to other things like formation of further degradation and oxidation to things like FAPI. And in the case of the 5-methocytosine, it can be oxidized here to form thymidine glycol. And again, here you can actually detect this. And this here, this particular type of modification results in mutations of which GC to AT transitions. Lipid peroxidation. Even though we can, under oxidative stress, we can modify certain components of DNA, one of the chief targets for oxidative damage is actually lipids. And how lipid peroxidation occurs is in 2 steps. One is the initiation, where you oxidize a lipid to just a simple lipid radical. Now, lipid radicals will react actually with oxygen in the form of the hydroperoxide radical, which then can actually react with another lipid here to form a lipid radical and lipid hydroperoxides here. And these lipid hydroperoxides can actually keep further generating these oxide radicals or peroxide radicals, which can further oxidize additional lipid molecules here, perpetuating in a propagation type mechanism. In organic chemistry, we call these propagation reactions, these radical reactions here. So that's one of the reasons that only a small initiation here can result to this type of propagation into a large number of molecules being oxidized. And a lot of decomposition products that you get out of this that you can detect are alkanes, MDA, 4-hydroxynonenal, and 8-isoprostane. And you can actually buy kits to measure many of these different types of reactions. If you look in what's happening in the membrane, and in the case of the membrane here, we have these alkene bonds here. They get oxidized here, react with oxygen here, and you can get this propagation to occur, and this ends up with the destruction of the membrane. So this is very bad, and we'll talk a little bit about vitamin E and how vitamin E can actually play a role, and other antioxidants can play a role in preventing this type of membrane damage. But lipid oxidation is not always bad. Again, everything has its place and time, and here, these are the eicosanoids. The eicosanoids start with arachidonic acid here, and arachidonic acid can get oxidized into a variety of different compounds, which are actually active. PG2 is one of our favorites from COX-2, and that mediates pain. That's why we take things like Vioxx, or I take diclofenac a lot because I hurt myself a lot. So, so oxidation is not necessarily bad, but when it's uncontrolled, like in the membrane, it can actually cause cellular damage. Is there any questions? Yeah. No, it's used to sterilize, right? Well, hydrogen peroxide can because it'll induce platelet aggregation as well. But there's a lot of factors. Platelet aggregation is, because NO is the converse of that. NO actually prevents platelet aggregation, but there are a number of different things. And hydrogen peroxide is also mediated by the, I'm going to get this wrong, by the MMPs, but also by, I forgot which carcinogen as well, too. But hydrogen peroxide generally is thought to just oxidize and sterilize the area. I think that's, I don't know that, yeah, I think that's more of a sterilization. I mean, although hydrogen peroxide does, I think, weakly induce platelet aggregation as well. So usually what happens when you have a wound, usually what happens is you see this cascade, immune cascade, and first it uses hydrogen peroxide and HOCl eventually, but that's your sterilization mechanisms. The ROSs are also signaling everything else, including the carcinogens, to start platelet aggregation. And it turns out NO is the shutoff valve. So eventually down the way, NO comes in through these higher levels of NO. It's sterilized. It has its own cascade of sterilization. But then that starts shutting down, inducing the wound healing response, like VEGF, things like that. Talk a little bit about that at the end. They didn't give me enough time. You think I run this thing, right? I'm the chair, right? No, they didn't let me talk about it. Said I have to talk about the chemistry. I apologize. I understand that this is a lot thrown at you. What I'm more interested in is that you hear the terms. You know, you can always ask me, but you can actually go in and start reading redox papers. You have the handouts so that you can actually go back. Oh, I think this is what this means, and then if you're interested in this, we can talk about techniques and things. The other guys' lectures are beautiful. You'll love them. So, anyways, here, tyrosine. Tyrosine modifications is another thing. If you have an oxidant and you have a tyrosine dimer here, that's actually fluorescent. You can detect that. That's an interesting way. As I mentioned before, tyrosine radicals can react with NO2 to form nitrotyrosine, and they also can be hydroxylated as well. So tyrosine can undergo a variety of different modifications. Proline. Proline can be oxidized to 4-hydroxyproline. This is interesting because prolohydroxylase. We always think of prolohydroxylase these days as the regulatory factor for HIF-1 alpha. So there's a proline, critical proline, that the prolohydroxylase simply has an iron complex that reacts with ascorbic acid and oxygen. And ascorbic acid is actually very important. And oxygen oxidizes the proline here and then targets it for ubiquination. So you're constantly producing HIF. If oxygen is there, it's being consumed. When there's no oxygen there, then this reaction can occur. The proline's there, it stabilizes the molecule, and HIF gets upregulated. But conversely, prolohydroxylase is also very important because if you don't have prolohydroxylase, your collagen is not stabilized. That's why you get scurvy with lack of fruits and vegetables. So these types of reactions here are more than just simply oxidizing things. Carbonyl formation here. You can oxidize different carbonyl formations. These can result in different peptides. There's a variety of different kits here which you can detect carbonylation formation in different types of proteins. Sulfur. Of the redox targets, sulfur is one of the big ones. I mean, metals and sulfurs. That's where you start first with many of these redox reactions. Methionine here can be oxidized here by an oxygen insertion here to form methionine sulfoxide. If you actually oxidize this with a radical, this will actually decompose to produce a number of different products which you actually can detect. Cysteine, intracellular antioxidants like glutathione. That's critical. If you've ever depleted the cell of glutathione, Terry told you about some of the mechanisms, you actually open the cell up to a lot of different types of stress, we'll call it. Chemical stress, oxidative stress. And so it's critical here. And why that is because the sulfur can scavenge a number of different reactive species. Even though it forms these sulfide radicals here, they can react with another sulfur and form these disulfides here. They can, if they're in isolation here, they can react with oxygen here, and that actually ends up making these higher ordered sulfoxides here. So in the presence of large things like glutathione, you can actually go to a mechanism here that results in these disulfides that the body can handle. Or you can get this sulfinic acid here, which is just SOH, which then can actually rapidly dimerize to SSR. But when the oxidants go this way here, you can actually get these higher states of oxidation of sulfur. And there's actually enzymes that can detect it. I'm sorry, antibodies that can detect this. But this generally represents an oxidative stress type situation. So let's turn to nitric oxide, certainly one of my favorite molecules. I got to NCI, and there were 70 papers on nitric oxide. And they were thinking, well, this is actually produced in the body, and nobody kind of believed it. But it's turned out since 1989, this has turned out to have almost 200,000 papers on the subject. They've given one Nobel Prize out. They're actually talking about another. And there isn't a single tissue in your body that does not have nitric oxide in it. Nitric oxide is a ubiquitous regulator. But it is a stable free radical. You can buy a bottle of this. You can call it Aldrich, or you can buy a bottle of this. As long as it doesn't see oxygen, it's fairly stable. And its redox chemistry is very, very important. This actually took friends of mine, myself and some collaborators, a long time to understand, because there's a lot of myths out there about nitric oxide. Since nitric oxide is a component of cigarettes, smoke, and it's also a component of smog, why are we producing this nitric oxide to regulate fundamental mechanisms such as blood pressure? Well, it turns out it's got a unique property. NO, it's very hard to oxidize NO to NO+. Now, NO+ is a nitrosonium ion here itself is actually fairly, it's fairly reactive. But NO can make nitroxyl here. And what we published here was the reduction potential here was very, very high. It's so high that to get to HNO here in the body, in the mammalian body, there really isn't any enzyme that can reduce that. There is in bacteria. Pyruvate dehydrogenase would be one. But there are no mammalian enzymes that can actually convert NO to nitroxyl. So NO sits in this well here. It's actually fairly stable in this well. And so it can't just simply pick up an electron from, like oxygen can, from the mitochondria. NO cannot easily be oxidized. And so it's actually fairly stable by itself. NO reacts with metals to form a metal nitrosyl complex. And actually, if you look at 90% of all the reactions of nitric oxide in the body, it's due to the fact that it binds to soluble guanylyl cyclase, which is a heme protein, which regulates conversion of GTP to cyclic GMP. NO can react with radicals here. And in fact, when NO reacts with lipid radicals, it protects the membranes from the propagation here of lipid peroxidation. And this radical-radical coupling also is part of the radiation-induced DNA radicals, the DNA radicals, which actually is why NO can actually enhance hypoxia radiation. And some of the reactions of NO are some of the fastest known in chemistry. So, but this is all under more hypoxic conditions. The one thing NO is that NO in the presence of oxygen auto-oxidizes ultimately in the presence of water to nitrite. And in the gas phase or lipid layers, this is a very unique reaction, is that two NOs have to react with oxygen to form this nitrogen dioxide. And I'm actually have to caveat this. I gave this course last time 2 years ago, and one of the guys was a computational biologist, Tao Ping, and he told me, I don't think so. So he went back, and actually I may have to change this. We haven't published a paper on this, but this may not just simply go to NO2. But anyways, the NO2 can react with NO to form N2O3. N2O3 plus water gives you nitrite. So it's a very simple reaction here. And the other thing, too, you have to be aware of, if you're working with NO, it does produce an acidic solution. A lot of people in the beginning of the days would dissolve all this NO up, and they have the NO oxygen, they put this bolus NO in there, and all go, and all of a sudden they have all this acid, and they wonder why their cells are dying. So you have to be a little careful of that. And in the aqueous reaction here, and this is something we've been talking about in our group for a long time, is we don't think there's really, we can't detect nitrogen dioxide, and we're still working on the intermediates. But the bottom line is the NO plus oxygen reaction does result in N2O3. And I hate to be, it's just in this last year we've had to reevaluate the actual mechanism of this reaction here. So, but what we do know about this is why the chemical kinetics here and why it's very important. This is a strange reaction because I have to, I'm dating myself here, but in 1976 I took freshman chemistry and we were doing kinetics. And my teacher, who was a gas phase smog person, said that the NO-oxygen reaction is very complex. And it's unique because it has this second order dependence in the rate limiting step. And you don't often see this in reactions. But the second order dependencies kind of explains why we can use nitric oxide as a critical signaling molecule. If you don't have nitric oxide, if you do knockout mice, they get sick really fast, mostly cardiovascular disease. So Mother Nature, at least in physiologically, uses NO, but in an aerobic environment, it should go away. Well, what this second order dependency means is that the half-life changes with NO concentration. So for example here, if you have 1 micromolar NO concentrations, it has a half-life of about 13 minutes. But if you go to 100 micromolar, it has a half-life of 8 seconds. So what's interesting about that is that you can have a cell producing NO here, but as it diffuses away from the cell here, it dilutes. And its lifetime actually gets longer and longer. The other thing is, since NO is also used in antibacterial and antipathogen responses, nitric oxide can be produced very high concentrations in the cell. It exponentially increases all those reactive nitrogen species through the NO-oxygen reaction. And when it diffuses away from the cell, again, its lifetime actually increases, but the amount of reactive nitrogen oxide species from the auto-oxidation reaction dramatically reduces. So that's why the unique, just simple chemistry of the NO-oxygen reaction explains a lot about why NO is actually used in many physiological conditions. Any questions about that? I haven't explained that in a couple of years. So when we were trying to understand nitric oxide, we developed this concept called the chemical biology of NO. Now, NO has a lot of different possibilities of reacting in biological systems, so how do we sort this out? So we actually tried to understand based on how fast reactions occur and what concentrations of NO would be required to see those types of reactions. And so we did this, and NO can react with heme proteins, like guanylate cyclase. Again, this requires only nanomolar amounts of NO. NO is scavenged by oxyhemoglobin, so it can't just travel all over the body. It's a big sink. It's like a black hole for nitric oxide. But when you actually have in places such as macrophages, you get this increase here. You get NO2 and N2O3. You get, when you actually increase the concentration here, these are what we call indirect effects. And these indirect effects here can occur in cells like activated macrophages. NO can react with superoxide to form peroxynitrite, which is a powerful oxidant unto itself, yet in the presence of excess oxygen, converts it over to NO2 and N2O3. So you actually have this kind of buffer here, and each one of these molecules here can actually react with different types of, different macromolecules here. So by dividing this up into direct effects and indirect effects, this requires sub-micromolar levels of NO, and indirect effects require concentrations that we estimate between 300 and 1 micromolar and above. And actually, we've determined that the concentration of NO in a microenvironment of a cell can be achieved at that level, even in humans. And of course, here, there's an emerging story here, nitrite and nitrates. But nitrite is a product, but that can be oxidized by peroxides back to NO2. And so this divides, on this side of the equation, you have nitrosative stress, and on this side, we favor oxidative stress. So this is kind of our general roadmap to try to understand the chemistry in the context of biology. So the direct reactions are NO with hemes. That's your classic one, soluble guanylate cyclase. Oxyhemo complexes here plus NO makes methemoglobin and nitrate. That's a very important scavenging reaction. Metallo-oxo species here, these iron oxo species are produced during the Fenton or peroxidase reactions, can react with NO to form nitrite. And these are all very fast reactions, and they require nanomolar concentrations. The Haber-Weiss reaction we talked about here, which can actually make hydroxyl radical, which can react with a variety of different things. NO can react with that. But more important, we think that NO reacts with superoxide to form peroxynitrite, which prevents this re-reduction here of the iron 3 back to iron 2. So NO actually is an antioxidant. NO also has antioxidant properties here. It's counteracting some of the basic chemistry here of lipid radicals. You form these hydroperoxyl radicals here. They can react with NO here. And this breaks this chain propagation steps. And NO can react with also the alkoxide radicals or the hydroperoxide radicals here. And again, break this chain of lipid peroxidation. And we've shown this in several different papers back over 20-some years ago. The nitric oxide was a powerful antioxidant. In fact, the way we're starting to see signaling from ROS versus NO is they actually tend to be antagonistic. Not always, but they tend to be antagonistic. And so the indirect chemistry here is when we actually react NO with either oxygen or superoxide to generate things like N2O3, which is nitrisation, which can result in nitration of different products. You can induce double-stranded DNA strand breaks or lipid peroxidation. Nitrisation chemistry from N2O3, you actually can form RS and Os. And nitrosamines, you can form secondary nitrosamines, which some have been reported to be carcinogenic. But one of our first papers with Larry Kiefer is we showed that N2O3 reacted with some bases in DNA, caused deamination here, and resulted in mutations which were consistent with transitions. There are other targets of N2O3, like S-nitrosothials. We discovered, along with Cronky et al., that zinc fingers were also susceptible to N2O3 back in the 90s. N2O3 can react with tyrosines, and even lysine, and even tryptophan. In this case here, many enzymes have been shown to be modified by S-nitrisation reactions. A number of proteins, such as PARP, the estrogen alpha receptor, and even some of these metalloprotease, which results in activation. So on this side here, these degrade the zinc fingers. These zinc thiolates and metalloproteases are actually activated. So it doesn't necessarily mean that these reactions don't necessarily mean degradation. Tyrosine, we've actually found out recently here, this is a work by Lisa Ridenour, the modifications here by N2O3 and nitrisation of critical tyrosines result in activation of CD63, which we think is promoting ER-negative breast cancer, or promoting poor prognosis in ER-negative breast cancer. Lysine, ligase, IGF, I think that was one of the first things that we did with Terry. And you can modify tryptophan, but there aren't really any examples yet. I mean, chemically you can do it, but I don't think there's anybody shown any examples of that yet. In this case here, the oxidation of NO2. NO2 thiols are critical for this. You form these thiol radicals here, which then can actually decompose into the disulfide ligands. NO2, NO-oxygen reaction can produce NO2. Also, formation of the peroxynitrite through addition of CO2 and NO can actually generate NO2 as well. Nitrite oxidation by things like MPO or any of the peroxidases can form NO2. NO2 can oxidize tyrosine to the tyrosine radical. You have the tyrosine radical can either form a dimer here, as Bert Meyer's group has shown, or it can react with NO2 to form nitrotyrosine here, which you can actually pick up with an antibody. So the indirect reactions here, the formation of things like NO2 and N2O3, you have nitrisation or oxidation or nitration. This is just your basic summary slide of what these indirect chemistries mean. You can react with superoxide and you can get back to NO2 through through peroxynitrite here, which can be actually converted over N2O3. So it really depends on the flux of this, these two ions, NO and superoxide. Nitrite under acidic conditions. And the reason we think that this is very important is if we look in the phagosome, phagosome is a very acidic environment, and nitrite from nitric oxide can actually be dumped into the phagosome. And when it's done that, you actually can produce N2O3 and NO2 simultaneously, which actually has antimicrobial effects. But what is really emerging like I said, I only have about 2 minutes left, but we are actually now starting to decipher all this chemistry. And if you want to read some more articles, I can give you some more articles on this. But if you want to try to understand this chemistry in the context of signal transduction, we've actually, yeah. Oh. It's actually the redox environment itself, I would say. The acidic environment, usually we're talking about pHs 4.5. You can get some of those acidic, 6.5 that you find in hypoxia or ischemia. But most of it's the redox environment. And so it's how much NO is actually being produced. So the levels, that's a really important question. The levels of NO are really dictating what the targets are. And the targets change. So the chemistry with the indirect effects are usually, like here, we usually about 50 nanomolar and below. I almost say this is exclusively metal reactions with guanylate cyclase. All the RNS stuff cannot occur. But as we raise the concentration of NO, NO gas and oxygen, right, are lipid soluble. So the reactions concentrate in the membranes as opposed to the cytoplasm. And so what we start to see here, and that's a really important question here, is about 300 nanomolar, we start getting activation of membrane-bound proteins, RAS, EGFR, SARC. And these activate things like AKT and ERK, and these induce, at least in our cancer models, cancer stem cell markers like CD44, COX2. So this actually, RNSs promote migration, they promote, which is good in wound healing, bad in cancer. They promote things like chemoresistance. They promote cancer stem cell markers. And so in this level here, because it's targeted in the membranes, those thiols in the membranes, this level here, we call this the metastasis, induces cancer stem cell-ness. But what we really, if you look at this in context of entire biology, this level here is really the wound healing level. If you look at any wound healing model or fibrosis models, you'll see a lot of the same players here. And when you start getting above this, you start inducing things like P53. This is the stress levels. This is when now those chemical reactions are occurring that are starting to produce a lot more RNS ubiquitously. And one thing that we've learned is the chemistry is elegant, but it's not random. When we've been working with the chemistry for 20 years, and I always think of the signal transduction pathways that's being influenced in the biological systems. The chemistry initiates it, but it's actually amplified by the biological system. And that's the most important thing about redox biology, is the amplification of the targets. So here we have specific nitrization of membrane, and this is a regulatory mechanism here. But when we start getting a lot of different molecules, nitrosative, we induce nitrosative stress at these levels here. In fact, we've measured markers for levels that would be equivalent to up to 5 micromolar flux for 24 to 48 hours. In fact, those markers are in human diseases. And down here, all this here, you have direct effects, which involve guanylate cyclase, things like angiogenesis are all induced by this. Also regulation of the cardiovascular system. Oh, I guess that's my end of my show. So the bottom line here is, I'm sorry, we went through a lot of chemistry here today. I promise it gets better. But one thing is the chemistry is important. All right, the kinetics are important to determine what your molecular marker is. But one thing, even while we've been teaching this course, you know, you'll read about everything is bad, but it's not. It's how those chemical interactions happen and they hit the biological targets. Biology is not just sedentary. It's not like throwing a bomb. It's the amplification of this chemistry here biologically, which actually determines the phenotype. Probably should have better food. Anyway, sorry.
Any questions for Dave?
Not too many. I got to go home and study government. My daughter is in AP government. She said, so, anyways. Yeah. Yeah. Right. Oh, I don't know that actually, but no, alkaline. I can probably tell you. I only have one bad habit. I wish it was beer, but it's not. I drink Diet Pepsi, which is probably as acidic, right, Terry? She's been trying to break me. No, but it's known as acidic. But that's a lot more complicated because the one thing we didn't mention here is the inorganic chemistry of redox biology is also the chemistry of the microbiome. And so when you're changing an acidic diet that you're talking about, you're actually influencing microbiome. Hydrogen sulfide, hydrogen gas, right? 40% of your gut gases is hydrogen sulfide. H2S, CO, nitric oxide, nitrates, nitrites are all metabolized in your gut microflora. So the your acidity is probably the interaction of your oral and gut microflora to selecting out the right bacteria. That's what that probably is. Has probably nothing to do with what, you have too many buffers to get, you know, if you drank a soda, that pH does not translate into your brain. But it does affect your microflora. Or microbiota, I guess is what, they don't like microflora, I found out. So.
Too much acidity, cavity.
Good treat. All right, thank you. We'll try to be more interesting next time.